series online. The Department of Studies in Biotechnology, University of Mysore is organizing a lecture series of alumni of the department and as well the university. So already 5-6 lectures, the product of the department they have delivered, and I thought that this lecture is going to help at least minimum of 5-6 life science departments. Professor Ranganath is well versed with the genetics and the genotype and phenotype through omics and overview as a title he has given. And this lecture is going to help the microbiology, zoology, botany, biotechnology, and genetics. So all these departments, biochemistry, they are going to benefit out of this lecture. So I requested Professor H. Ranganath is busy schedule. He agreed, readily agreed. Whenever I ask, he will not say no. And then I thought that, sir, you deliver a lecture, the message will go to students. Because the department this year admitted 15 national level students from JNU stream. So they are offline. So they are expecting more teaching from the university. Anyhow, we know that the online teaching, whether it's effective or not, that we cannot take it as a debate now. But the online teaching should be benefited because there is no alternate for the students now. University is doing in that way. All the teachers of the university, they are taking a task of the online teaching every day. Teachers are doing. Including that, we have thought that some of the alumni are there. They are working in the very good laboratories in the country as well as abroad. So in this way, I requested Professor Ranganath to deliver a lecture on genotype to phenotype through omics and overview. And he agreed. For this occasion, I requested our Honorable Vice Chancellor, Professor G. Hemant Kumar, sir, to inaugurate this lecture. And he readily agreed, sir. Sir, I welcome you for this alumni lecture of Professor Ranganath. And also with us, guest of honor, our very dynamic and young leader of the Registrar of University of Mysore. And he is also readily agreed to attend. He is always, because he is a social work man. Social work is common subject for all. Ultimately, the science or any technology or any output of the science, it has to reach the society. That Shivappa has to help out to reach the society. So he is taking role of all the lectures and the webinars, role and involving in the academic endeavor, and he is showing his interest. That's a good sign of the university to take off further because the NAC is coming. We have to show that the offline teaching and how many lecture series we have organized, that we need the account on that one. And the registrar is going to take the stock of these things of last few years, almost 1.5 years we are offline. So students are going to benefit it. Shivappa also agreed, and for this webinar, I welcome you, sir, for this wonderful lecture of the Professor Ranganath. I also welcome my colleagues, especially Dr. Umesh. He is the coordinator of MHRD. Luckily, the department has got the extension of MHRD for another 5 years. The DPT has supported for us, and we have given the new proposal for that one, and it is agreed. And newly, the responsibility has taken by the Umesh to take the responsibility for another 5 years to run the HRD program. They are giving some contingency, student fellowship, and we admitted 50 students, and the authorities have readily agreed and given the 15 seats also. And our number is increased 51 students in this year. Biotechnology is the 51 students, of course. The laboratory and practically we are thinking this difficult, but without our knowledge, these 15 members are admitted so that it has gone up to 51. Otherwise, it was around 30. But this 15 number has increased 51, but Vice Chancellor has given the new building for us, new facility, new laboratories, new classrooms, so that we are not facing any problem, so that we are managing with the national students and they are also happy. And in this one, the Umesh will take care about the MHRD program. And Professor Aparna, the senior professor, she is going to take the charge as a chairman in the few days come future, and she will take the responsibility of the department. Another 10 days, my term is going to be completed as chairman, and Aparna is going to take the department responsibility. And also Professor Kiri is also senior professor, and Dr. Geeta and Dr. Lokesh, they are all together. They are the young generation and the new line of the leaders. They are going to take the department. They need your blessings to take the department as we have taken earlier with the international and national programs. So I welcome all of you, and also I welcome Professor Dandin. I thought that many of them, Ranganath, we have sent the Ranganath lecture webinar to everybody, and many of them are, I think, how many? What is the number now? 70, and the students are still, because students, as they thinking that they are logging like a classroom, so that it is going to increase. And on behalf of the university and the department, I welcome over personally. Professor Vishwanath is there, Professor Dandin is there, Professor Malini is there, all other chairmen of the department and other professors, other intellectuals and scientists. Welcome for this webinar, sir. Once again, I welcome Professor Ranganath, Professor Bharti Salimat, Professor Meva Singh, and all of them, Professor Ramachandra, everybody is there. And it is a one group of scientists who are joined in the hand with the Professor Ranganath. And Professor Ranganath, once again, sir, I welcome you for this webinar, sir. And I thankful on the behalf of the university and on behalf of my colleagues and the department. We are grateful to you to accepting our invitation to deliver the lecture, sir. So thank you once again for one and all, and welcome all of you for this webinar. Thank you very much.
So very good morning to you all. Our senior Professor H.A. Ranganath, Professor S.R. Niranjan, all the colleagues of the biotechnology department, and many students and researchers and many colleagues are online. So this Department of Biotechnology is doing very excellent work since its inception. So we were also the part of computer science, was a part of biotechnology. So I started the career there. So I used to see the Professor Prakash and Professor Shetty and how they used to work and us in the beginning. So all other teachers are also following the same path, how it was developed. So it is one of the excellent department. So they didn't ask anything. I only gave them 2 buildings to what we call to come up with that. So anyway, with the infrastructure currently, so as Professor Niranjan told, this again, this DST, this DBT has helped to what we call continue for 5 more years this program. I congratulate all the faculty and the chairman of the department and the coordinator. This is a very alumni lecture series is a very important thing because most of the alumni will be useful to our young generation. So many departments are conducting this type of seminars and lecture series. So during last lockdown period, our university has conducted more than 135 what we call lecture series during the webinars during that time. So this is also one of the essential lecture series, and also Professor H.A. Ranganath is our own man. Always he supports us, he guides us whenever what we call something he sees in the about the university. Always he calls me and tells about the things. So he's always a guiding spirit for us. Today his lecture is there. So I genotype and also genotype to phenotype and through what we call omics, an overview. This is the topic of the lecture. This is the current topic which is required. I am very happy to inaugurate this lecture on this day. Thank you very much. All the best to you all.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ ಸರ್. ನಮ್ಮ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಕಾರ್ಯಕ್ರಮ ಆಗ್ತಾ ಇರೋದು ತುಂಬಾ ಸಂತೋಷ ಅನಿಸ್ತಾ ಇದೆ. ವಿಶ್ವವಿದ್ಯಾಲಯಗಳನ್ನ ಕಟ್ಟಿದಂತ, ವಿಶ್ವವಿದ್ಯಾಲಯಗಳಿಗೆ ಕಾಯಕಲ್ಪ ಮಾಡಿದಂತ ಬಹಳ ಈ ಒಂದು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಇತಿಹಾಸದಲ್ಲಿರ್ಲಿ, ಬೆಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿರ್ಲಿ, ಮತ್ತು ನ್ಯಾಕ್ನ ಸಂದರ್ಭಕ್ಕೆ ಬಂದಾಗ ನ್ಯಾಕ್ನ ರೂಪುರೇಷೆಯನ್ನೇ ಬದಲು ಮಾಡಿ ಅದರ ಒಂದು ಶ್ರೇಯಾಂಕವನ್ನ ನ್ಯಾಕ್ನ ಒಂದು ಹೇಗೆ ನಾವು ಮಾನದಂಡವಾಗಿ ಮಾಡಬೇಕು ರಚನಾತ್ಮಕವಾದಂತ ಬದಲಾವಣೆಗಳನ್ನ ಮಾಡಿದಂತ ನಮ್ಮ ವಿಶ್ವವಿದ್ಯಾಲಯದ ಪ್ರೀತಿಯ ಹಿರಿಯ ಪ್ರಾಧ್ಯಾಪಕರಾದಂತ ಪ್ರೊಫೆಸರ್ H.A. ರಂಗನಾಥ್ ಸರ್ ಅವರು ಇವತ್ತು ಅಲುಮ್ನಿ ಲೆಕ್ಚರ್ ಸೀರೀಸ್ ಅಲ್ಲಿ ಮಾತಾಡ್ತಾ ಇರುವಂತದ್ದು ಬಹಳ ಸಂತೋಷದ ವಿಷಯ. ನಮ್ಮ ಬಯೋಟೆಕ್ನಾಲಜಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ನಮ್ಮ VC ಸರ್ ಈಗಾಗಲೇ ಹೇಳಿದ ಹಾಗೆ ವಿಶ್ವವಿದ್ಯಾಲಯದ ಹೆಗ್ಗಳಿಕೆಗೆ ಹಲವು ಕಾರಣಗಳನ್ನ, ಹಲವು ಶ್ರೇಯಗಳನ್ನ ಕೊಟ್ಟಿದೆ. ಸೊ ಕೆಲವೇ ವಿಜ್ಞಾನದ ವಿಭಾಗಗಳು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕೀರ್ತಿಗೆ ಪಾತ್ರವಾಗಿವೆ. ಸೊ ಅದರಲ್ಲಿ ನಮ್ಮ ಬಯೋಟೆಕ್ನಾಲಜಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಕೂಡ ಒಂದು. ಅದೇ ಅನೇಕ ನಿಮ್ಮಂತ ಹಿರಿಯರು ಮತ್ತು ಈಗಿರುವಂತವರು ಎಲ್ಲರೂ ಕೂಡ ಬಹಳ ಅವಿರತ ಶ್ರಮಿಸಿ ವಿಶ್ವವಿದ್ಯಾಲಯಕ್ಕೆ ಒಳ್ಳೆಯ ಹೆಸರನ್ನು ತಂದುಕೊಡ್ತಾ ಇದ್ದಾರೆ. ನಾನು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಪರವಾಗಿ ಇದನ್ನ ಅವ್ರನ್ನ ಅಭಿನಂದಿಸ್ತೇನೆ. ಮತ್ತು ಈ ಬದಲಾದ ಸಂದರ್ಭದಲ್ಲಿ, ಬದಲಾದ ಕಾಲಘಟ್ಟದಲ್ಲಿ ವಿ ನೀಡ್ ಟು ಕ್ಯಾಚ್ ಅಪ್ ವಿತ್ ದ ಚೇಂಜಸ್ ಅಂತ ಏನೇ ಹೇಳ್ತೀವಿ, ಹಾಗೆ ಈ ಕೋವಿಡ್ ನಮ್ಗೆ ಹಲವು ಪಾಠಗಳನ್ನು ಕಲಿಸಿದೆ. ಈಗ ನೋಡಿ 82 ವಿದ್ಯಾರ್ಥಿಗಳು ಆನ್ಲೈನ್ ಅಲ್ಲಿ ಕಾತುರತೆಯಿಂದ ಇದ್ದಾರೆ ತಮ್ಮ ಲೆಕ್ಚರ್ನ ಕೇಳೋದಕ್ಕೆ. ಸೊ ನಾವು ಕೂಡ 1.5 ವರ್ಷದಲ್ಲಿ ಟೆಕ್ನಾಲಜಿನ ಬಳಸ್ಕೊಂಡು ನಾವು ಸುಮ್ಮನೆ ಇರಬಾರ್ದು ಅನ್ನೋ ರೀತಿಯಲ್ಲಿ ಬಹಳ ಒಳ್ಳೆಯ ಪ್ರಯತ್ನಗಳಾಗಿದೆ. ನಮ್ಮ ಈಗಿರುವಂತ ಕುಲಪತಿಗಳು ಒಂದು IT ಇನ್ಫ್ರಾಸ್ಟ್ರಕ್ಚರ್ ಅವರ ಒಂದು ನೇತೃತ್ವದಲ್ಲೇ ನಮ್ಗೆ ಕಳೆದ 15 ವರ್ಷಗಳಲ್ಲಿ ವಿಶ್ವವಿದ್ಯಾಲಯ ಅನೇಕ ಬದಲಾವಣೆಗಳನ್ನು ಕಂಡಿದೆ. ವಿಜ್ಞಾನ ಭವನ, ಇಲ್ಲಿರುವಂತಹ ಟೆಕ್ನಾಲಜಿ, ಆಮೇಲೆ ಸೈನ್ಸ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ನಲ್ಲಿರುವಂತಹ ಇನ್ಫ್ರಾಸ್ಟ್ರಕ್ಚರ್, ಇವೆಲ್ಲವೂ ಕೂಡ ನಮ್ಗೆ ಈ ಕೋವಿಡ್ ಸಂದರ್ಭದಲ್ಲಿ ಬೇರೆ ಯಾವ ವಿಶ್ವವಿದ್ಯಾಲಯದಲ್ಲಿ ಇದೆಯೋ ಇಲ್ಲೋ ಗೊತ್ತಿಲ್ಲ ನನಗೆ, ಕೆಲವು ವಿಶ್ವವಿದ್ಯಾಲಯಗಳಲ್ಲಿ ಇಲ್ಲ, ಅದಂತೂ ಗೊತ್ತಿದೆ. ಆದ್ರೆ ನಮ್ಮ ವಿಶ್ವವಿದ್ಯಾಲಯ ಇದರಲ್ಲಿ ಏನು ಕೊರತೆ ಆಗದ ಇರೋ ತರದಲ್ಲಿ ಹಿಂದೆ ಇದ್ದಂತ ಕುಲಪತಿಗಳು ಮತ್ತು ಈಗಿರುವಂತವ್ರು ಅದನ್ನ ಮಾಡಿದ್ದಾರೆ. ನಾವು ಕ್ಯಾಚ್ ಅಪ್ ವಿತ್ ದ ಚೇಂಜಸ್ ಅಂಡ್ ಟೈಮ್ಸ್. ನಮ್ಮ ವಿದ್ಯಾರ್ಥಿಗಳು ಕೂಡ ಅನೇಕ ಪಾಠ ಪ್ರವಚನಗಳನ್ನ ಆನ್ಲೈನ್ ಅಲ್ಲೇ ಕೇಳ್ತಾ ಇದ್ದಾರೆ. ನನ್ಗೆ ಗೊತ್ತಿದೆ ಕೆಲವು ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಅದು ಅನುಕೂಲ ಸಿಗ್ತಾ ಇಲ್ಲ ಬಿಕಾಸ್ ಆಫ್ IT, ಇಂಟರ್ನೆಟ್ ಮತ್ತು ಕೆಲವೆಲ್ಲ ಆರ್ಥಿಕ ಕಾರಣಗಳಿಗೋಸ್ಕರ. ಅದನ್ನು ಕೂಡ ನಾವು ಬಗೆಹರಿಸಲಿಕ್ಕೆ ಪ್ರಯತ್ನ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಈ ಒಂದು ಸಂದರ್ಭದಲ್ಲಿ ಈ ತರ ಒಂದು ವೆಬಿನಾರ್ಗಳನ್ನ ಮಾಡಬೇಕು, ಪ್ರತಿಯೊಂದು ವಿಭಾಗದಿಂದ ಕೂಡ ನಾವು ಕನಿಷ್ಠ ವಾರಕ್ಕೆ ಒಂದು ಮಾಡ್ಬೇಕು ಅಂತ ಹೇಳಿ ಮಾನ್ಯ ಕುಲಪತಿಗಳು ನಿರ್ದೇಶನ ನೀಡಿದ್ರು. ಅದರ ಹಿನ್ನೆಲೆಯಲ್ಲಿ ಎಲ್ಲ ವಿಭಾಗಗಳು ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಸೈನ್ಸ್ ವಿಭಾಗಗಳಂತೂ ತುಂಬಾ ಚೆನ್ನಾಗಿ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಕೆಲವು ನಮ್ಮ ಸೋಷಿಯಲ್ ವರ್ಕ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಅಲ್ಲೂ ಕೂಡ ಹಲವಾರು ಮಾಡಿದ್ದಾರೆ. ಒನ್ ಆಫ್ ದಿ ಬೆಸ್ಟ್ ಪರ್ಫಾರ್ಮಿಂಗ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಅಂತ ನ್ಯಾಕ್ ಕೂಡ ಹೇಳಿದೆ ಲಾಸ್ಟ್ ಟೈಮ್ ಇದರಲ್ಲಿ. ಹಾಗಾಗಿ ನಾವು ಬದಲಾಗಬೇಕಾಗಿದೆ. ನಮ್ಮ ಅಧ್ಯಾಪಕರು ಕೂಡ ಮನಸ್ಥಿತಿ ಅದೆಲ್ಲ ಬದಲಾಗಿದೆ, ವಿದ್ಯಾರ್ಥಿಗಳ ಮನಸ್ಥಿತಿ ಬದಲಾಗಿದೆ, ಬದುಕನ್ನು ನೋಡುವ ಕ್ರಮವೇ ಬದಲಾಗಿದೆ. ಸೊ ಹಾಗಾಗಿ ಈ ಬದಲಾದ ಸಂದರ್ಭದಲ್ಲಿ ಈ ತರದ್ದು ಒಂದು ಲೆಕ್ಚರ್ ಸೀರೀಸ್ ನ ಬಹಳ ಅರ್ಥಪೂರ್ಣವಾಗಿ ಹಮ್ಮಿಕೊಂಡಿರುವಂತ ನಾನು ಬಯೋಟೆಕ್ನಾಲಜಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ನ ಮತ್ತೊಮ್ಮೆ ಅಭಿನಂದಿಸಿ, ನಮ್ಮ ವಿಶ್ರಾಂತ ಕುಲಪತಿಗಳು ಈಗ ತಾನೇ ಇನ್ನೇನು ನಿವೃತ್ತಿಯನ್ನು ಅವರಾಗೆ ಅನ್ಕೊಂಡ್ಬಿಟ್ಟಿದ್ದಾರೆ ನಾನು ನಿವೃತ್ತಿ ಆಗ್ತಾ ಇದ್ದೀನಿ ಅಂತ. ಬಟ್ H. A. ರಂಗನಾಥ್ ಸರ್ ಹೇಗೆ ನಮ್ಮ ಜೊತೆ ವಿಶ್ವವಿದ್ಯಾಲಯದ ಜೊತೆ ಇದ್ದಾರೆ, ಅದೇ ರೀತಿಯಲ್ಲಿ ಇವರು ಕೂಡ ನಮ್ಮ ಜೊತೆ ಇರ್ತಾರೆ, ಸದಾ ಇರ್ತಾರೆ ಅನ್ನುವಂತ ಮಾತನ್ನ ಹೇಳ್ತಾ, ಬಹಳ ದೊಡ್ಡ ದೊಡ್ಡ ಪ್ರಾಧ್ಯಾಪಕರು ಈಗ ಈ ವೆಬಿನಾರ್ ಅಲ್ಲಿ ಭಾಗವಹಿಸ್ತಾ ಇದ್ದಾರೆ. ಎಲ್ಲರಿಗೂ ನಾನು ನಮಸ್ಕಾರಗಳನ್ನು ಹೇಳ್ತಾ, ವಿಶ್ವವಿದ್ಯಾಲಯದ ಪರವಾಗಿ ಮತ್ತೊಮ್ಮೆ ಅಭಿನಂದನೆಗಳನ್ನ ತಿಳಿಸ್ತಾ ನನ್ನ ಮಾತನ್ನು ಮುಗಿಸ್ತೀನಿ. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ.
So I welcome Professor Chandranaik is also here with us, and I forgot to welcome him. So Professor Chandranaik is also joining with us for the webinar. Now I request, in the interest of the students, normally Professor Ranganath, they don't want to tell these biodata for the young generation. So I told Umesh to take up that part to introduce Professor Ranganath for the new generation of students in the campus and as well the outside. Dr. Umesh.
Niranjan, one request. Let us not lose time in doing these things.
2 minutes
ಎಲ್ಲರಿಗೂ ಗೊತ್ತಾಗ್ಬೇಕು,
ಎಲ್ಲರಿಗೂ ಗೊತ್ತಾಗ್ಬೇಕು ಸರ್. ಬೇಕು
ಯಂಗ್ಸ್ಟರ್ಸ್ ಒಂದು ಮಾಡ್ಲಾಗ್ಬೇಕು.
ಯಂಗ್ಸ್ಟರ್ಸ್ ಗೆ ಒಂದು ಬೇಕು ಸರ್. ಇಲ್ಲ ಅಂದ್ರೆ ಅವ್ರು ಯಾರೋ ಬಂದ್ರು ಹೋದ್ರು ಅಂತೇಳಿ ಅವ್ರು ಏನೇನು ಹೊಟ್ಬಿಡ್ತಾರೆ, ಯಾರೋ ಗೊತ್ತಾಗಲಿಲ್ಲ ಅವ್ರು ಯಾರು ಅಂತ. ಹಂಗಾಗಿ ಗೊತ್ತಾಗ್ಬೇಕು ಸರ್ ಅದು. ಹೇಳಿದೆ ನಿಮ್ ಕೂತಿದ್ದು ಗೊತ್ತಾಗಿಲ್ಲ, ಲಾಗಿನ್ ಆಗಿಲ್ಲ. So I welcome Professor Chandranaik once again for this webinar. Introduce him.
Good morning to all of you. Respected Vice Chancellor, Professor Hemant Kumar sir, Registrar Professor Shobha sir, and our esteemed teacher Professor S.R. Niranjan, former Vice Chancellor and the Lieutenant Chairman. It gives me an immense pleasure to introduce today's speaker, Professor H. A. Ranganath. Professor Asan Annegowda Ranganath was born on 16th June 1948. He obtained his PhD in 1975 from the University of Mysore, and he has postdoctoral experience in the laboratories of Germany and UK. Professor Ranganath has served the University of Mysore for over 3 decades, 1975 to 2008, in different capacities. Since the early 1970s, Ranganath has made original contributions in the area of genetics and evolution, and has published extensively in reputed journals. With the Drosophila as model organism, he pioneered the studies on the experimental evolution in India. Research projects of Professor H. A. Ranganath are funded by INSA, UGC, DBT, DST, DFT Germany, and other funding agencies. With the support of his colleagues, as well as the university, Professor Ranganath was instrumental in establishing 2 national research facilities at the University of Mysore, namely Drosophila Stock Center, and Unit on Evolution and Genetics, funded by DBT, Government of India, and DST, Government of India, respectively, as well as of a new program, new PG program, namely MSc in Genetics. Professor Rangnath has received recognitions and awards for his contributions to the evolutionary genetics. He is the elected fellow of all the three science academies of our country, namely Indian Academy of Sciences, FASC, National Academy of Sciences, FNASC, Indian National Science Academy, FNA, and fellow Karnataka Science and Technology Academy, FKSTA. Other awards of Professor H. Rangnath, Professor was awarded the Indian National Science Academy Young Scientist Medal 1979, UGC Career Award 1986, Golden Jubilee Award of University of Mysore 1994, Professor Y.T. Tatachari National Award for Science 2003, Aryabhatta Award 2008, Sarya Vishesharaya Vignan Puraskar Award 2010, Dr. Raja Ramanna Award 2017, Sri Venkateshwara University has conferred on Professor H. Rangnath the honorary degree of Doctor of Science. Professor Rangnath is the Vice President of Indian Society of Evolutionary Biologists. After serving as Vice Chancellor, Bangalore University, 2006 to 2008, Professor Rangnath sir was the Director, National Assessment and Accreditation Council, NAC 2008 to 2013, an autonomous institution of UGC. He was a visiting professor for 5 years, 2014 to 2019 at IISc Bangalore. Also, Professor Rangnath was editor-in-chief of one of the oldest and prestigious international journal, the Journal of Genetics, published by Indian Academy of Sciences. Currently, Professor S. J. Rangnath is the Chairman, Board of Governors, IIIT Kurnool, visiting professor, Center for Human Genetics, Bangalore. Now he is recognized as the Distinguished Professor for Life of the University of Mysore. Professor Rangnath was on many committees on Government of India, such as UGC, MHRD, Planning Commission, DST, DBT, AICTE, NBA, QCI, CSRMTI, Science Academies of the country, such as INSA, IASC, KSTA, statutory bodies of the state and the central governments, etc. And Professor also in the committees of the government of Karnataka. Currently, Professor Rangnath is interested in the research topic, actively involved in the teaching, genetic and evolution, and issues concerned with higher education. With this very brief introduction, I welcome once again Professor H. Rangnath to this students and also University of Mysore. Thank you, sir. Over to you, sir.
Mr. Vice Chancellor, Professor Hemant Kumar sir, Mr. Registrar Shivappa sir, Professor Niranjan, all my colleagues of the Department of Biotechnology, and all the former colleagues who have logged in, and I could see the all original earlier faces of my good friends and ladies and gentlemen. Hemant Kumar, I can, whatever Niranjan said was a lie. Because I requested him, you invite youngsters to speak on such subjects. We senior most citizens with 70 plus, and we can only give the history, but not the contemporary issues. But he insists.
Number 90, sir. 90 means there is a lot of young generation is there. Only old people maybe 10 to 15. Other 80 members are young generation.
Okay. Anyway, sir, I will try. I'm not sure whether Niranjan has done the right decision for inviting me to deliver this talk. I will try to do my best. Thank you, Niranjan, for inviting me, sir.
Okay, sir. Thank you very much. Can I share the slides?
Yes, sir.
Can I start?
Slide
ಆಗಿಲ್ವಾ?
ಇಲ್ಲ ಸರ್, ಸ್ಲೈಡ್ ಶೇರ್ ಆಗಿಲ್ಲ. ಶೇರ್ ಕೊಡಿ, ಶೇರಿಂಗ್ ಕೊಡಿ. ಸ್ಕ್ರೀನ್ ಶೇರ್ ಕೊಡಿ.
DCM meeting ಇದೆ.
DCM ಆನ್ಲೈನ್.
Online. 12:30 to 2.
ಕಾಣ್ತಾ ಇದೆಯಾ?
ಇನ್ನೊಂದಪ್ಪ
ಸ್ಲೈಡ್ ಶೋ ಮಾಡ್ಕೊಡಿ. ಸರ್ ಕಾಣ್ತಾ ಇದೆ ಸರ್. ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಇಲ್ಲಿಗೆ ಬರ್ತೀನಿ.
So I have chosen it because Niranjan sir told me that students are the ones who have to target. Therefore, I have selected this topic so that particularly the first year and the second year MSc students may be benefited out of this. But when I saw on the guest list, for example, Professor Vishwanath, Professor Ramchandra, Professor Moline, and biochemistry for my colleague, Madam, all of them are and Dandin. Really, I'm not comfortable to speak of the subject because these are still they're very, very much alive in the subject. But anyway, I will try to go through this process of sharing my thoughts, what I learned from Gangotri. So whatever I shared today and whatever the praise in the introduction he praised me, Umesh, is all the credit goes to my alma mater, Department of Zoology and Manas Gangotri campus. But for this campus, but for the zoology department, we would have been a zero, sir. So I pay respect to my department. And whenever Malini writes, I usually call her and tell her, whenever I visit Mysore, my first visit would be my alma mater, that is Department of Zoology. So that's how the training at Manasa Gangotri and Zoology Department has shaped our career. And I'm extremely grateful to the campus and my teachers who trained me. So, sir, now I will go through the process of sharing some of my thoughts on this particular subject. So this is the topic I have chosen. And this is William Johansen, who gave us this concept of phenotype and the genotype. And at that particular point of time, that is, as you could see the date 1909, phenotype, he just meant the physical appearance of an organism. And by doing breeding experiments, he decided what would be the genotype. At that time, people did not know what genotype is. Only through breeding experiments, he tried to establish a correlation between what the phenotype, what it appears, and what would be the genotype. So that happened in 1909. Now, from 1909 to 2020, now during these years, 100 years, many things have happened in biology, and I will try to give an overview of the developments and the evolution in our understanding of the relationship between genotype and phenotype. Wilhelm Johansen was the man who started this journey, and still those concepts are very much alive in our discussion. And first few slides are just a recapitulation, what we have learned earlier, what we have studied, for example, phenotype, genotype, and the omics. So it is an age of omics, and finally I will try to bring a relationship between the classical concepts of the phenotype and the genotype. You know this was the working model of Gregor Mendel, and by using the phenotypes, the shape of the seed, color of the seed, etc., he did breeding experiments, and in all his breeding experiments, he looked at the phenotypes of the plant products and how they appeared in the parents and how they manifested in F1 and how they behaved in the F2. So for all this discussion, the central point was the phenotype. So by looking at the phenotype of the pea plants in the parents, in the F1 and the F2, he gave us the basic foundations of genetics, which all of you know, law of dominance or segregation or assortment. All these decisions, hypotheses were conceived by looking at the phenotype. What I'm trying to stress is genetics was born by looking at how the phenotypes, how the characters flow from parents to offspring. So as you know that for 3.5 decades, his work was not appreciated and studied. Only in 1900, 3 people independently rediscovered the Mendelian contributions, and it was reborn. So at that time, nobody knew what gene was there. Nobody knew what was gene. It was invisible structure, and it was a statistical concept. The behavior, the pattern of inheritance, etc. By looking at the phenotype, and not only just the phenotype, there are statistical ratios. That means whether the character appears in 3:1 ratio or 9:3 3:1 or so these statistical ratios used to tell us the behavior of these invisible structures. Invisible structure of the gene, the presence or absence of the gene was considered by looking at the phenotype and their frequencies. So during those periods, 1900 to 1940, the gene was a statistical concept. So by looking at the statistical ratios, people learned many dimensions of genetics. So after rediscovery of Mendelian principle, again by looking at the phenotype and the ratios, many more concepts, many more patterns of inheritance were learned, which were not enunciated in the Mendelian principles. For example, incomplete dominance or codominance, epistasis, linkage, even genome imprinting, etc. All these things were learned after 1900. And these are all, usually we say these are the extensions of Mendelian principles. Again, my emphasis is on all these things were learned by looking at the phenotypes. And through the breeding experiments, people started to imagine what will be the genotype which are giving such different phenotypes. So now, this is the equation. So genotype, they did not know what that genotype was, and we knew what the phenotype is, and absolutely there was no information to connect how these things are happening, how genotype is determining the phenotype. So this question mark continues till the end of the lecture, of course, in different forms, etc. So as you know, the syndrome of genetics, Drosophila. By doing breeding experiments, Morgan and his students, and particularly this fly, the normal fly has the red eye and the mutant fly has the white. This was a breakthrough in biology. This was a breakthrough in genetics. By doing experiments, Morgan and his team showed that this phenotype, white phenotype or the red phenotype, is due to a factor present on the X chromosome. So they knew the phenotype now, and also by studying the pattern of inheritance of the eye color in Drosophila, they could show that a particular factor is present on the X chromosome, and also they showed through the breeding experiment, the eye color, red eye color was dominant and the white eye color was recessive. So by looking at this dominant and the recessiveness of the fly, they decided the genotype. The genotype is this fly must be homozygous, a recessive character must be homozygous, and this could be a homozygous for the dominant color, or it could be heterozygous. Like that, the breeding experiments helped Morgan and his team to not only look at the phenotypes and also to decide the genotype. The genotype of this fly must be homozygous for W, and this must be homozygous or heterozygous for the dominant gene. That's how this fly, particularly the white-eyed fly in Drosophila, started the journey of pattern of inheritance, not only for Drosophila, even for the entire biology. So by doing such experiments, the genotype was written. W is the gene for the eye color. The white eye color, because it is recessive, that the genotype has to be homozygous. And when in this homozygous genotype, the eye color will be white. If there's a dominant gene, then it would have been red. So like this, so by doing such breeding experiments, the first level of relationship was established. That is, this is the genotype and this would be the phenotype. That is the starting point. Later on, hundreds of mutants of Drosophila were utilized to learn more and more about the genotypes and also more and more about the phenotype. And now, the next thing was, as I said, hundreds of mutant characters were seen in Drosophila. They were isolated particularly from Morgan's lab, and later on it was shared and spread to other labs in US and Europe. And people started to know how exactly these new phenotypes, new variations have come into existence. Red was the eye color. Suddenly white eye appears. All the flies had normal wings. Suddenly the wing becomes vestigial. So appearance of these variations in the phenotype. And now they wanted to know what are the causative factors which bring in these variations in the phenotypes now. Because these phenotypes are the ones which are helping us to learn the pattern of inheritance, etc. And they wanted to know the root cause for the emergence of these variations. And at that time, the concept was already there. Mutations are the ones which bring in new variations, new characters. But a question mark continues here because at that time, they did not know what is this mutation, but only they know the phenomena is a mutation which brings about a heritable change. To understand the structure of the process and the dynamics of mutations, you need to know the gene and its structure. See the question marks, and at that time, nobody knew what that gene, what that structure, what that mutation could be. So then only we can appreciate how the new phenotypes, new variations are going to be formed. So almost at the same time, when people are probing, they are trying to understand the mechanism of mutations, which is responsible for bringing new phenotypes, new characters. Almost at the same time, almost parallelly, see, Addington was studying a developmental biologist. He was looking at the development. See the date, 1942. When rediscovery of Mendelian principles have taken place, genetics has grown to understand more and more dimensions of inheritance, like codominance, linkage, crossing over, etc. During that particular time, the heydays of classical genetics, C.D. Wannington, he was studying the embryology, development. And his experience was this. The germ cell consists of, that is the zygote, consists of a nucleus, and at that point of time, it is a homogeneous cytoplasm. Nothing else was there. Of course, today, with modern techniques, you can show it is not homogeneous, but already there is cytoplasmic differentiation. So the germ, he considered germ cell is a homogeneous substance, and this germ cell gradually undergoes cleavage. Progressively, new structures are going to appear. He was a developmental biologist. New formation from which epigenesis was derived. That means from this single cell, many new organs, new structures come up, and that process was called as new genesis takes place. So here, the cells differentiate and organs form. From a single cell, you get varieties of phenotypes now, varieties of tissues, varieties of organs, varieties of organ systems, etc. So during development, from a single cell, he could record, he could demonstrate new phenotypes appear during development. Each system, it could be a vascular system or a reproductive system or nervous system, each one of them has a different phenotype. But not only different phenotypes, they are structurally, functionally different. Such highly differentiated phenotypes appear during development and differentiation. So Waddington was astonished to see how a single cell, from a single cell, which was a homogeneous mass of cytoplasm and one nucleus, what are the factors that bring in such new phenotypes progressively from single cell till the adult is born, progressively new phenotypes, new structures. So Mendel, Morgan and his workers, they were looking at the characters inherited from parents to offspring, new phenotypes, new variations, new mutations. Waddington was looking at the appearance of phenotypes during development and differentiation. So at this stage, I may be excused because there may be spelling mistakes, because when I type these things, because of distraction, etc., I might have committed a few spelling mistakes. I may be excused for this. So the new phenotypes, novel phenotypes, committed phenotype performing definite functions are appearing during development. So he was interested in this problem. So now at that stage, new phenotypes can come into existence because of two processes. One is mutation, another is epigenesis, that is the developmental profile during the development. But for both of them, what a mutation, people did not know. How epigenesis is responsible, what are the epigenetic events that take place during development, people did not. But the net result is mutations are responsible for new phenotypes. Similarly, epigenesis is also equally more important to bring about the normal development and differentiation. So the next challenge was to understand the mechanisms of these mutations, the processes underlying epigenesis, so that we can appreciate how new phenotypes are going to be formed. So during the story of my talk, I am trying to establish a relationship between genotype and phenotype, taking note of the developments in science, particularly biology, at different points of time. So what are, first let me take the mutations. These are heritable changes. And as you know, that till 1940, gene was an abstract entity. From only after 1940 onwards, people did couple of experiments, and then they realized what these genes are, and also they started to understand how so-called these genes control different characters or different phenotypes. That happened. These answers to these questions came up during these 3 decades, that is 1940 to 1970. So the first breakthrough was this. 1940 was the important date in biology. We call it as the birth of genetic chemistry. So because of many discoveries during this particular period, 1940 to 1950, the genetic principles and the chemistry of the substance were have to be amalgamated. And now people say that genetic chemistry was born because in 1944, Hershey, Avery, MacLeod, and McCarthy, they did experiments with bacteria and bacteriophages and the viruses. So till 1940, it was the heydays of Drosophila, heydays of maize plant. But after 1940 to up to 1970, 75, during these decades, the working material model system was the prokaryotes, the bacteria and viruses, and the bacterial transformation experiments, etc., conducted by these people showed that DNA is the hereditary material. That is, the gene. For the first time, gene assumes a characteristic feature of a chemical substance which is visible now. You can isolate a DNA, and the DNA is visible now. Till now, as I said, gene was an abstract entity, is an invisible structure. But after 1944, when once the hereditary material is recognized by these people, the gene has to be treated as a chemical entity, and that is deoxyribonucleic acid. So whatever you are, henceforth, whatever these people speak of gene structure and function, they have to speak in terms of DNA. So DNA is the genotype, and this DNA, this genotype, this gene somehow dictates the appearance of phenotypes. So as you know that man has about 3 billion nucleotides with 25 to 20,000 genes, and you know these things. So the DNA was recognized, and the next step was to see what the DNA is. And you know the chemistry of the DNA was worked out. It is made of purines and pyrimidines, and just the composition was not enough, because by knowing the composition, you cannot decide how this gene, or this genotype, this hereditary material, with only these 4 bases. These are the only components. These are the only 4 components, and the mystery of that particular point of time was, we say this is the hereditary material. All genes are made up of this, And the mystery or the challenge was to account for how this simple structure, entire genome made up of such 4 repeating units of 4 units, would give rise to such a diversity of life on Earth. So the important thing that has to happen immediately was to understand the structure of the gene, structure of DNA, because the emphasis from that day is to have a relationship between structure and function. So they need to have a structure for the hereditary material, and with that structure, they must be in a position to explain the functions that is expected of a gene, the functions expected of the hereditary material. So you know now that many people were in the race to resolve the structure, and these physicists, chemists, biologists, biochemists, all these people joined together and they gave us the famous structure of the gene, structure of the DNA. I will not go into the details. So after 1944, DNA was recognized as the hereditary material, and the next 8 to 9 years, intense investigations happened to understand the structure of the gene, and you know that's how physicists, chemists, biologists, biochemists, modeling, etc. All these people put their brains and developed a model, and even today, we go through this model for many of our discussions. So now we know the structure of gene, structure of the DNA, and now we have to relate how that structure changes. Whenever it changes, we get what we already called it as a mutation. And because of these mutations, new variations, new phenotypes would come. So the next challenge was to understand the process of mutations, types of mutations, etc., and how new phenotypes are going to be formed. So in simple terms, heritable changes are brought about by changes in the message of the gene. And as you know, that message of the gene is imprinted in the form of the sequence of these nucleotides in the DNA. And when such sequence is altered, by some process, a mutation will happen. And when such mutations happen in the genotype, you get new phenotypes. So particularly for my student friends, I have summed up a research of 10 years or a decade. People spend their lifetime to understand how these things happen. And in this talk, as I said, it's an overview, and new phenotypes can come up by the process of mutation. Basically, mutations point down to changes in the sequence of bases like this. So mutations, now they are related to the phenotypes. And now, as you have seen, you can classify the mutations based on the structure. It could be deletions, duplications, or transposable elements, substitution mutations, transitions, like that. Depending upon the depth of your knowledge, width of your knowledge, you can go on defining the mutations in different contexts. Similarly, you can look at the mutations in how it performs. Some mutations, we call it as a mutation has happened, but it does not perform any function. You call that as a loss of function mutation. Sometimes we call it as amorphic mutation. That means morphology is going, no, nothing will happen to that amorphic. Haploinsufficiency, frame shift, like that, gain of function. Of course, those who teach mutations, those who look at how these different mutations bring about new characters, an outline classification is like this. Some mutations are hypermorphs, some are antimorphs, neomorphs, etc. What I'm trying to do, when once they realized mutations are the causative factors for new phenotypes, more and more breeding experiments, more and more induction of mutations, etc. They dissected the various types of mutations that one can see in different life forms. So at this stage, again, Drosophila came back. Drosophila came back to tell us what is this loss of function mutation, what is this gain of function mutation, how these things happen. When once it was clearly established in Drosophila, other model systems also looked at it, and these mutations are not restricted, are unique to Drosophila. Such similar activities are taking place even in other model systems. So if you look at the effect of mutation on the protein, protein is one type of phenotype. Sometimes, you know, the synonymous mutations, non-synonymous, missense, nonsense, etc. Like that. As I said, depending on the width and the breadth of the knowledge a teacher or a student has, we can go on classifying mutations. Some mutations are there, neutral mutations, it may not affect the fitness. Sometimes there are lethal mutations which will change the fitness matter. So whatever it is. So mutations are the founding or the raw materials on which the new phenotypes are brought about, and the mutations could assume different dimensions. And now we know the structure. People realize the structure of the gene by 1953-54. And next question is how this gene, this DNA, this hereditary material determine different characters, different phenotypes. And they started their investigations, particularly again to start with bacteria and the bacteriophages. The activities of the nucleic acids or the hereditary material like replication, transcription, translation, recombination, mutation, repair, regulation, etc. So these are the different transactions of the hereditary material. So hereditary material, the gene is DNA, and molecular biologists, biochemists put together using bacteria and bacteriophages showed us. Even today, we use all these things when we teach and when we discuss. The foundations were laid. The foundations were laid for the dynamics, the enzyme chemistry, the process of replication in prokaryotes from 1940, 1950 to 1975. So all even today, the knowledge what we gained during those period is the foundation. And whatever things, when we started to look at the eukaryotes after 1975, we studied replication in eukaryotes, replication in eukaryotes, transcription in eukaryotes. So these are the things to start with in prokaryotes, and the same processes were resolved in eukaryotes. That type of extension helped us to learn more and more about the multiple facets of the hereditary material. So foundations were laid, and now these things, particularly replication, transcription, translation, or mutation, all these things are involved in deciding the phenotype. So the first step, they resolved the gene, hereditary material, the structure, and now the function. So there is whatever Watson gave the structure, there was a beautiful relationship between its functions. What are its functions? These are the functions. So even though Watson and Crick did not give us the mechanism for these functions, but he gave us the structure, with that model, one can explain all these activities of the hereditary. These are the expected functions of the hereditary material. That model gave by Watson and Crick. With that model, one can explain all these different things. So now I come back to the equation. Genotype, earlier I wrote WW gene, and now it is DNA. And now how this DNA is going to determine the protein. Protein is the first phenotype of the gene. Later on, and once the protein is formed, other things would happen. So the question mark continues. Earlier question mark was here. Now we know that it is the genotype, and that genotype is replaced by DNA, and this DNA is a particular phenotype. It could be a protein, and later on any other phenotype of your choice. So these are the three important molecules in biology: deoxyribonucleic acid, and proteins. Usually we call them as HOLYPOLYMERS. And when they were trying to understand the relationship between these polymers, central dogma molecular biology, all of you know that the information runs from one macromolecule to the other. DNA is a polymer, polynucleotide, and ribonucleic acid is another polymer, and the protein is another polymer, polynucleotide chain. So like this, the informational content runs from one polymer to the other. And this process, the first process is called as transcription. The second process is called translation. Again, I retain this question mark, because unless this happens, you cannot get a protein. You cannot get a phenotype. Protein represents a phenotype now. So it's like that. As I said, by resolving one step, some more questions are added to the next step. That's how science progresses. The discovery of one leads to many more questions, and when those questions were resolved, further questions could be asked. This is how the information runs from the gene, from the genotype to the phenotype. Of course, technology has come now. As I said, 3 billion nucleotides today. The genome sequence is available, and today robots can do the sequencing now. The entire genetic complement of the cell could be analyzed not by scientists, but by the robots, and now it can give you the entire genome of any person. In Bangalore, there are many companies who could give you a genome of any person at a cost of about less than $1 lakh. So that means earlier it was a challenge. Today it has become so easily available to people. And now the gene, the genotype, the DNA, and the genome is nothing but, and this genome, which is nothing but the entire complement of the nucleus, decides the phenotype now. So now, so far, I gave an overview of mutations which are responsible for new phenotypes. New phenotypes could be due to two reasons. One is mutation, another is epigenesis. So let me go through briskly, what that epigenesis means. Because epigenesis is as important as mutations now. Epigenesis and the emergence of new phenotypes, mechanisms. So let me define what the epigenetics or epigenesis means. Epigenesis, appearance of new phenotypes from the same gene. If you recall what Waddington said, from a single germ cell, trillions of cells are going to be formed. Different tissues, different organs, different systems are going to be formed with new phenotypes, new functions, new structures. So epigenesis means inheritance of altered gene expression. Because of altered gene expression, the new phenotypes are going to be formed. Here, please, the emphasis is on inheritance of altered gene expression. The mutation, the emphasis is on altered structure. Structure of the gene itself is to be altered, wherein the sequence of the nucleotides are going to be changed. In contrast to that, here, whenever a new phenotype appears because of epigenesis, you could see here, new phenotypes, new formations formed just by changing the expression. By changing the expression of the same gene, new characters are going to be formulated. So here, without altering the structure of the nucleotide sequence. So gene structure remains as it is. It has not been changed, but some regulatory events would decide what type of action that would perform and what type of function it would lead to a different phenotype. So it is not a mutation, but it is epigenetic changes are just due to differential expression of the gene. So therefore, the altered phenotype is not due to mutation, because we have defined mutation has an heritable change, and that happens because of changes in the nucleotide sequence or the structure of the gene. In contrast to that, here what is happening, the structure is unaltered, but the expression pattern is changed because of regulatory elements. That is epigenetics. Epigenetics deals with the differential expression of the same gene, differential manifestation of the same gene, giving new phenotypes. So let me compare for students. Mutation is an heritable change due to changes in the structure of the gene. When this happens, the informational content is altered, and therefore it is straightforward. New phenotypes are formed, new phenotypes are revealed just because the changes in the information. In contrast to this, we call it as epimutation. This is also heritable. This is also heritable. And this is brought about by changing in the gene function. So the gene expression is reversible. The same gene which is performing at some stage during development will not function after a certain time. And it is eclipsed. It becomes non-functional. For some time it is non-functional. Again, a regulatory event will bring that particular gene back to action. So epimutations or epigenetic changes are reversible. Within the same individual, for some time, the gene is performing a function. The same gene, because of a different decision during development, the same gene which was performing hitherto becomes non-functional. When it becomes non-functional or a different function, a new phenotype will form. And these two, activity and inactivity of a gene, is reversible as long as the structure is not changed. Structure remains same. The regulatory events would decide whether gene remains in a functional mode or in inactive mode. So it is reversible. All these things happen in the absence of DNA sequence, and new phenotypes are formed. Just a comparison. So here, the crux of the problem is this. Both are heritable changes. Both are heritable changes, but here the heritable change can be easily explained because it is the structure, the gene itself is altered, and that gene is possibly passed on from parents to offspring. But whereas here the structure is not changed, but even then it is heritable. So even today, many models are available to explain the epigenetic changes, the mechanism, how it is heritable when compared to the concept of heritability in terms of mutations. So like this. So now basic anatomy was understood, basic philosophy was appreciated, epigenetic changes do bring about new phenotypes, but structure is not altered. That means information remains same. Retaining the same genetic information, the same nucleotide sequence. However, how to explain, how to account for the appearance of new phenotypes. So now again they looked at this. Most of the work is done in eukaryotes. So when they looked at the chromatin, of the eukaryotic chromosome, you know the structure of the chromosome, chromatin, etc. Nucleosomes are there, and they looked at there must be something happening in these nucleosomes, so that the expression is going to be altered. And in this nucleosome, what you see is the nucleotide bases, histones, and non-histone proteins, H1, H2, H3, H4 linker proteins, and non-histone proteins, even RNA. So all these things are available, are packed in the nucleosome. So entire chromatic chromosome is nothing but a string of nucleosomes, and these nucleosomes have this component. Here, this is the core of the gene. This is the core of the gene. The informational content of the gene is determined by this nucleotide sequence. Right? So in an epigenetic change, the sequence is not altered. The sequence is not altered, thymine remains thymine, or cytosine are gone. So now the challenge for the people was to account for heritable change without changing the informational content. So these were the candidates. So now you know this the chromosome organization. These are the nucleosomes, and this is the DNA, and then it is going to be condensed. The nucleosomes are organized, histones, etc. You know all that, and finally a compact chromosome is going to be formed. So now they have to explain how this structure of the chromatin, without changing the genetic content, how to explain, how to account for the formation of new phenotypes. So the candidates for focus were DNA, histone, and the protein. Sorry, histone and the non-coding protein, RNA. And of course, this is the methylation. Of course, the nucleic acid, the nucleotides the bases undergo modifications. And these bases are going to be modified by the addition or the deletion of certain material. For example, here, this is the cytosine. There are enzymes who will tag on this cytosine with a methyl group, right? If this cytosine is decorated with methyl group under the influence of this DNA enzyme, then the structure of this DNA is going to be altered. That means the methylated cytosine has an impact on the structure of the DNA. So this addition of the methyl group makes this particular gene to be repressed. It becomes inactive because this particular methyl group condenses the DNA. When once the DNA is condensed, it is not accessible for transcription. For the transcription to take place, the double helix has to open up, and it has to be approachable by these enzymes which take part in transcription. When once it is methylated, this DNA becomes compact and it is not accessible for transcription. Therefore, it becomes inactive. Cytosine remains a cytosine, still it pairs with guanine. So informational content is not changed, but the presence of methyl group in the cytosine will make it condensed and it is not accessible. And as I said, as long as this is present here, this gene remains inactive. When once by another enzyme, demethylase is another enzyme that if that enzyme comes into picture, this methyl group is removed, then it becomes cytosine, not a methyl. As long as it is present, it is methylated cytosine. When it is removed, it becomes cytosine. The presence of this makes it inactive. The release of this CH3 methyl group makes it functional. So this is epigenetic change. So somebody during development, some regulatory mechanism is deciding whether methylase enzyme is active or demethylase enzyme is active. This is a family of enzymes. So if the methylase enzyme is active, it adds this. If a demethylase enzyme is as a player, it removes it. So very crudely, what I have said is the methylated cytosine is very common, and that decides whether the chromatin material is accessible for transcription. So one is like that. The bases could be modified, and depending upon the type of modification, this gene can become repressed or it could be activated. And the next candidate is the histones. Histones are present in the nucleosomes, right? And you know that the histones are small basic proteins, and it's extremely conserved in evolution. That means if you look at the histones of Drosophila, Caenorhabditis, or human, or anybody, the histones remain as it is. That's why we say if genes coding for the histone family genes are extremely conserved. The next point for us for discussion is, if you look at this nucleosome, the histones wrap, and the tail of the histone protein is a polypeptide. The tail, which has about 20 amino acids, is hanging. And now these 20 amino acids of the histone, which is part of the tail, is extremely accessible for modification. So again, I have to tell you, histone is a protein, a polypeptide, and there are genes which code for histones. Histone gene codes for histone protein, and once the translation is over, first transcription, then translation, once the histone protein is formed, immediately the amino acids present in this particular region, amino acids present in the tail region of the nucleosome are subjected to post-translational modifications. When you learn protein synthesis, you will see that the the nascent polypeptide chain, which is formed during translation, will be subjected to post-translational modifications. Only when post-translational modifications are effectively achieved, that protein becomes functional. So post-translational modifications are very crucial. And now, as far as this is concerned, histones are a product of the histone gene. Histone protein is produced, and in this polypeptide chain, the first 20 amino acids which are present in the tail region are extremely amenable for modifications. Depending upon the type of modification, depending upon the type of nature of modifications these amino acids undergo, again, the structure of the chromatin is going to be altered. For example, in the histone tail, the amino acids serine, threonine, tyrosine undergo phosphorylation. There it was methylation of the cytosine. Here, in the histone tail, in these 20 amino acids, the amino acids serine, threonine, tyrosine undergo phosphorylation. Similarly, lysine, arginine are methylated or acetylated, ubiquitinated, like that. I'm just giving you the list. These modifications happen after translation, and these modifications are very crucial because depending upon the modification that these different amino acids have undergone during translation process, post-translational modifications, what we call it, these histone modifications are referred to as marks, and are the sites, are the recognition sites. Recognition for what? The chromatin material has to be recognized by proteins when a transcription takes place. Enzymes have to interact with the chromatin material. The chromatin has to be opened up. So there must be a strong interaction between the chromatin-associated proteins and these marks. Depending upon the marks here, nothing but the modifications. These modifications decide what type of proteins it would interact. So depending upon this interaction between the modifications and the protein, the decision is whether the gene remains functional or not. So at one stage, modifications of the nucleotides would decide whether the gene remains functional or not. Now at the second level, modifications, alterations in the amino acids of the histone also contribute to whether the gene remains in a functional status or not. So all these days, last 20 years almost now, like genetic code, we have histone code now. Because the histone code varies from one tissue to the other. Because different genes are active in different tissues of the body, right? So in order to make sure that the respective required genes remain active in different parts of the body during development and differentiation, these genes are going to be marked, and that marking is called the code. These modifications alter the histone interactions with DNA and other nuclear proteins. So the histone code is very unique. Histone code is very unique to different tissues and organ systems of our body. So by looking at the histone code, you can see whether the gene is in a repressed state or an activation state. So as we see, as you all know, that the same genome is present in all tissues of our body, but different genes are active in different parts of the body, and also in time and space, the expression varies. The variations in the expression of the genes in time and during development, maybe at different points of time or in the different regions of the body, depends on the modifications of the nucleotide bases, and the modifications of the histone proteins of the nucleosome. And particularly, this is called a histone code, parallel to the genetic code is universal. Genetic code remains same in all tissues and cells, whereas histone code varies from one tissue to the other, because that forms the bottom line to explain differential gene expression. already people have mapped. Oh God. So that slide is not coming up. People have mapped the histone code in different tissues of the body. So by looking at the histone code of the nucleosome in different parts of the body, which is now available, it is available in the public domain, and one can see the difference in the gene expression in different tissues, which is responsible for different phenotypes. So one is the nucleotide base modifications, and next is the modifications in the amino acids of the histones. The third candidate, which is responsible for epigenetic change, is the non-coding RNA. So is also present on the nucleosomes. Of course, it's not a permanent component of the nucleosome. Depending upon the status of the activity of the chromatin, status of the activity of the gene, this non-coding RNA appears and disappears now. So non-coding RNA also decides the functional status profile of the gene. For example, a very common example I could tell you, in females, in mammals, one of the X chromosome is inactivated. One of the X chromosome becomes a bar body, a lionized hypothesis. So here, there are many more things happen for that lionization. Many more molecular events do happen. But one important thing for our discussion today is the X chromosome, which becomes inactive, which is inactive, transcriptionally inactive, is under the strong influence of two species of RNA molecule. They are non-coding because this RNA is not coding for protein. It's only this type of RNA. Its only role is to make that one of the X chromosome becomes inactive. This is one, as I said, this is only one step in this process of inactivation. There are many more multiple steps to decide which chromosome becomes inactive. As far as the epigenetic change is concerned, as long as this non-coding RNA, that this species of non-coding RNA is present on the X chromosome, it remains inactive. Once this non-coding RNA is removed by an experimental means, that the same chromosome becomes active. Similarly, in Drosophila, opposite thing happens. In Drosophila male, single X chromosome is there, whereas in females, two X chromosomes are there. In spite of this difference, in spite of the difference between male and female, male has one, female has two X chromosomes. In spite of this quantitative difference, the quantum of X-linked gene products, only one copy of the X-linked gene is present in male. There are two copies in the female, yet the quantum of X-linked products remains same. This is called dosage compensation. And here in this Drosophila, the single X chromosome, which is hyperactive, is again under the strong influence of a RNA molecule. So epigenetically, single X chromosome in Drosophila becomes hyperactive. Again, there are many more players to decide the hyperactivity. But one important player is the non-coding RNA. Whereas here in mammals, the dosage compensation is achieved by inactivating one of the X in females. So in both the cases, hyperactivity in Drosophila, inactivity in mammals is due to some varieties of RNAs. So all these are epigenetic changes. When once the RNA is present, the X chromosome becomes inactive, and when that RNA is removed, it becomes active again. So like this, I've given you a few examples to show that epigenetic changes are brought about by modifications in the bases of the nucleotides or the amino acids in the histone proteins and the interaction with the RNA. All these are epigenetic changes, temporary changes, and this is purely under the regulatory process of the developing individual. So now, because of these things, a new field has come up in the last 20 years, that is epigenomics. That means epigenetic modifications of the genetic material. So during development and differentiation, like genomics, today we have epigenomics. Genomics, the outcome of genomics was to understand the nucleotide sequence of the entire genome. Now epigenome is targeting to understand epigenetic modification, that is modifications in the nucleotide bases, modifications in the amino acids of the histones, and other changes in the genetic material. So epigenomics is trying to understand more and more about the epigenetic modification. This is very crucial because these are the modifications that decides the normal development and differentiation of the individuals. So because of the importance, today, like Human Genome Project, HGP, today, in the last 15 years, we have 2 more projects have come up, internationally collaborated projects, Human Epigenome Project, and a consortium. This collaboration aim is to focus on all areas of epigenetics, to bring about changes, how these epigenetic changes result in new phenotypes. So they are trying to understand methylation patterns and profiles in human genes in all major tissues, and their diseases. So a normal liver or a malignant liver, a malignant tumor in an ovary or a normal ovary. Like that, the current research is focusing on disease phenotype now. And most of the diseases have a different phenotypes, and people are trying to understand through epigenetic, what are the epigenetic changes that are introduced in an ovary which is being malignant, or in an ovary which remains normal. So this is an international consortium. They're trying to give us more and more information about the formation of new phenotypes through epigenetic changes. And the first consortium meeting took place during 2015. And they prepared the roadmap. Roadmap is the epigenomic signatures, broad spectrum of human tissues and cells. So we see the language now becomes signatures, epigenetic signatures in different tissues of the human cells during development. During development, how these transitions take, because a single zygote is undergoing transitions to give rise to different stem cells, tissue-specific stem cells, organ-specific stem cells, etc. These are crucial transitions. So now the epigenetic signatures, epigenetic modification are being analyzed, dissected, and it is being mapped during development. Human Genome Project 2001 gave us the nucleotide sequence. Now the Epigenomic Mapping Consortium is giving non-genetic modifications. So this consortium is giving us non-genetic modifications of the genome, crucially determine which genes are expressed in which cell type and when. So these epigenetic modifications are the bottom line to explain which genes are expressed in which cell type and at one point of time. When all these things are regulated by appropriate events, it's a normal tissue. If a disease, if a pathology comes in picture, then they can see what are the mistakes that have happened in the epigenetic change. So epigenomics deals with non-genetic modifications. Human Genome Genomics gave us the static nucleotide sequence of the genome. This static nucleotide sequence of the genome becomes functional only when it undergoes non-genetic modifications. And these non-genetic modifications are being mapped today, both in a normal tissue and an abnormal tissue. And only recently, or last month, I think, today it is July, last month, an important paper came working on yeast. They have analyzed about 80 genes in this paper, Nature Communications. Methylation, cytosine methylation is much more frequent than any other modification. Among epigenetic changes, methylation is, people thought methylation is very, very common, very, very dominant. Earlier it was like that. Earlier people used to say methylated cytosine could be treated as the fifth nucleotide in the DNA. We have 4 nucleotides. The methylated cytosine is so common, and therefore people are tempted to say methylated cytosine could be a fifth nucleotide. But now this work, which was published only last month, they say the much more common alteration, epigenetic alteration, is the histone 3 lysine 27. So the methylation of the histone 3 in the 27th region is much more common than the earlier accepted the methylation process of the DNA. So like this, even today, more and more information is pouring in to explain the epigenetic changes because epigenetic changes have assumed greater importance today. So now, as I said, from a homogeneous cell, 30 million cells are going to be formed, and each of these 30 million cells have received the genomes from the father and the mother. But if you look at the diversity of phenotypes during development and differentiation, it is astonishing. How genes are switched on or switched off in different cells at different stages of development. This is the biggest challenge even today. Today, Epigenetic Consortium, basic biology and disease-oriented research, right? It is a completely applied. DNA candidates are looking at this, histone modifications, chromatin accessibility, small RNA transcription stem cells, primary ex vivo tissues, normal counterparts, etc. Like this, the consortium, Epigenetic Consortium, is looking at all these things in normal and abnormal tissues, and they have mapped it. And they are trying to relate. They are trying to relate to the diseases that are seen in different individuals. For example, you know this. This is how the development, finally the adult tissue is formed. From this homogeneous embryonic stem cells, the blastocyst, then the fetus, then the baby. And now people have studied the epigenetic changes that are taking place during this development and differentiation. For example, of course, this is the metaphor given by C.D. Waddington. All of them are homogeneous cells, and you can see these are highly differentiated tissues and organs. He derived it in 1950s. So this is a valley, this is a hill, and only to say that it is if this cell takes this valley, this becomes one tissue. The same clone, the fate will be different depending upon the regulation process. So now what has happened is, these are the embryonic pluripotent stem cells, differentiation, and then all these different highly specialized structures. So all these things are happening because of epigenetic changes. It's a pluripotent and undergoes change, and you can see that different tissues are formed. For example, you take the hemopoietic tissue. This is a stem cell, hemopoietic stem cell now, first division, second division, and finally, highly differentiated cells are there. Highly specialized cells are there. All of them have the same genotype, but the phenotypes are different. The structure is different. The functions are different. Now, the beauty of epigenetics is from the pluripotent embryonic stem cell, this is derived. People have mapped the changes that have taken place here. And now, from this particular stage, from the bone marrow, they are tracing the epigenetic changes that are happening at different stages. So now if there is a problem at this stage in the epigenetic modification, some type of disease would come. If there is a wrong mistake in the epigenetic change, some other disease would come. So like that, now people have the information how these new phenotypes have come up and what are the normal epigenetic changes that have taken place. And whenever there's a mistake, and they could map what is that mistake, and because of that mistake, what type of disease that can happen. So these are the committed progenitors of the different levels and highly specialized. So now this is a disease recognition oriented research now. All these days it is a academic interest to know the epigenetic modifications, but today epigenetic modifications in this lineage can be mapped and it can be related to a disease. See, these are all different phenotypes. My entire talk depends on the genotype. The genotype, and this is the phenotype. And you see, varieties of phenotypes have come up, and the bottom causative factor for these phenotype changes is epigenetic change. These are not mutations. Similarly, the nervous system. So again, the same story, you could see varieties of cells. Varieties of cells in the muscular system. And today, the epigenomes of 127 human tissues and cell types are available. 127 human tissues and cell types are available, and starting from embryonic stem cells, induced pluripotent stem cells, that means highly differentiated cells. They were reversed because of the experimental means. You can reverse highly differentiated cells back to the pluripotent and see what are the changes, and the adult tissues. So embryonic stem cells, adult tissues, and now you can have IPS, induced pluri. That means from the highly specialized cell, you can reverse the process to get back the pluripotent and see what are the changes. So at all these three levels, from 127 tissues, information is available in the public domain from the healthy individuals, and also with people with cancer, Alzheimer's, etc., etc. So information is available to know what are the epigenetic changes that have taken place in a normal individual that lead to a breast cancer, or of course in breast cancer already BRCA1, BRCA2, all these have been analyzed, and people are looking more and more about this. See, is Julian Huxley, 65 years ago, that is 1956, at a time when people have not understood the mechanism of epigenetics, etc. He said, I shall use epigenetics as meaning the science of development process. So he said, epigenetics becomes the bottom line for developmental biology development. That's what we are seeing today. He expressed it in 56. That is almost 65 years ago. He conceived that epigenetics is extremely crucial for biology, and that's what we have seen with all this development of science and technology. Huxley went on to describe epigenetics in a manner which we typically understand in the 21st century. Encompass the complexity and the multiplicity phenomena, defining how genes and environment. So all these things he said in 1956. I only bring this quote just to impress upon you how visionaries these were. He was basically an evolutionary biologist and basically a good philosopher, and he could, after Waddington, and then he realized the importance. And now in the last 2 years, an important breakthrough has happened in molecular biology. This is called developmental barcoding of the whole mouse via, that last 2 years back, CRISPR technology got the Nobel Prize. I will not go into the details. I will just give you how technology is helping us to learn more and more about the relationship between the genotype and the phenotype. You have the DNA profiling, DNA barcode for individuals, DNA fingerprinting. Similarly, like DNA fingerprinting, today with this technology, you can have genetic barcodes tracing cells back to a single cell origin. As I said, the entire development starts from a homogeneous cell, then differentiation takes place. So hematopoietic stem cell, there is only one cell. And from this single hematopoietic stem cell, a bunch of cells come out performing different functions. So now, as I said, people are trying to map the epigenetic changes that are happening during this path of development. Now, this technology, what we call genetic barcode development technology, has refined that. Of course, it was the Harvard University happened. They published in 2018, and more publications have come up. This is the stems to all life as a single cell, then divides to 8 cells, 26 billion cells, etc. The grand challenge of developmental biology is to trace. Now, it is like a phylogeny now. The phylogeny of these 26 billion cells. What is the origin? What is the differentiation? And how things have happened? We can create a phylogenetic history of each cell line today with this particular technology. Of course, now this is a new type of RNA that is called homing guide RNA, HgRNA. Of course, you can refer any book, you will get it. And this RNA is used, this RNA is used to develop this technology. In brief, I will not go into the detail. See, these HgRNA molecules are engineered in such a way that when it is, the Cas9 and CRISPR enzyme is present, what happens is the HgRNA, homogeneous RNA, will guide this enzyme, This RNA this is how the interaction takes place in simple terms. This HgRNA will guide this CRISPR-Cas9 enzyme to cut the DNA, this sequence. It will damage this sequence. So this is a suicidal mechanism. This RNA is provoking this enzyme to cut its own sequence, right? And then when this sequence is damaged, the cell will try to repair it. This broken HgRNA will be repaired because the repair mechanisms. During this repair process, mistakes do happen. Mistakes do happen, and those mistakes are nothing but mutations. So these HgRNA cut and the repair mechanisms vary from one tissue to the other. So people are following the type of damage that has happened to the HgRNA and what are the, when the repair takes place, what are the mutations that have happened, and these mutations that have happened in this sequence are being mapped. And these mutations that are happened in liver tissue or a vascular system or a nervous system will be different from one another. So people are using this particular, it's a very in simple terms what I have said, mutations that happen in the Hg sequence under the influence of Cas9 is being mapped. And because of this, the barcode of the liver tissue is different from the barcode of hemopoietic tissue or a nervous tissue or a muscular tissue. Like this one. Because like DNA profiling, we can have the barcode now. So this during development, these are the changes that have happened from the embryonic stem cell. So we can say one barcode is different from the other. Like that, at Harvard, the entire barcode of development and differentiation is mapped in the mouse system. Of course, now they were extremely successful in the mouse, but they are trying to replicate. So the importance of this discovery is being able to record cells continuously over time is a huge milestone in development. So at every cell division, at every cleavage, you can follow what is happening. That promises exponentially increased understanding of our how single cell grows to form an adult individual. All these days used to have theoretical models. Today, because of this barcoding, you can show that if there are 100 varieties of cells in the body, there will be 100 varieties of barcodes in an individual. Or if there are 35 different types of, already I said, 124 cell types are available. Epigenetic modification. For each of these 124, you have a different type of barcode. Now, this is the most, if applied to disease models, it could provide new insights into how diseases can emerge. So that means if what we know today, the normal barcode of a normal lung, or when lung fibrosis takes place, what would be the difference? Like that, it is, as I said, that the research is moving towards understanding more and more about the basics of biology and also its correlation with the diseases. So now finally, the genome may be present if the genome has to have a say, epigenetic changes have to take place. That's why many people feel that epigenome brings genome to life. Genome alone can't do anything. So new phenotypes, new things can happen only through epigenetic changes. So this is how new phenotypes can come up, either through mutations or through epigenetic changes. So all of you, one can ask you a question: how many epigenomes we have? How to analyze the epigenomic data? Can we keep our epigenome healthy? Because many of the epigenetic changes that are happening in a body is because of so many. People have worked out what are when a cardiovascular disease comes up, what are the epigenetic changes and what are the causative factors. When the lung is suffering from problem, how the smoking has affected, how the carbon molecules, how the radiation. So one of the themes in some of the labs is that keep the epigenome healthy. So that epigenetic, we know what are the epigenetic changes, and these epigenetic changes have to be taken care of. So now, this is the main thing I wanted to share. I'll go briskly now. The DNA to protein, and now in between, we have got the epigenotype now. Earlier, the equation was simple, from DNA to protein, a genotype to phenotype. Now, if the genotype is to give you a phenotype, it has to pass through a step called epigenotype. And we know now what are the modifications that are taking place in the epigenotype. So most of these things you know now, the central dogma of molecular biology. Now, the next, if the epigenotype is formed, the next thing is it has to send this information to RNA through transcription. And I will not go into the details of the mechanisms, you know that. Transcription, the transcriptome is transcribed to RNA, and this is the cartoon to say what things are happening during the transcription. And the important part is this. Transcriptome is a complete set of RNA molecules, right? And again, post-transcriptional modifications. And routinely, it's happening in many labs. And the important thing is this. Today, you could see here varieties of RNA molecules. These RNA molecules, mRNA is the one that is coding, but there are varieties of RNA molecules which are non-coding, and they play a very crucial role, including the homing guide RNA, what just now I described. Like that, varieties of RNA molecules are there in the biology of the cell, which will influence the vital functions of the DNA. Regulation of transcription. So, like genomics, like epigenomics, today you have transcriptomics. And again, people have developed softwares, and people could provide information about the transcriptomics. And again, you could see here, they have, these are the people, and any one of you who has some information about the RNA transcription process, etc., you can contact them, and you can also contribute to the growth of this particular technology, and also can add your contribution to this thing. That means it's growing very fast. So next, RNA is ready, and now the translation, the protein. You know all these things. I will skip these things. So the polypeptide is going to be generated. As I said, post-translational modifications, the proteins. And you know this, we have about 20,000 genes, but we have about 1 followed by 6 so many proteins. It is because of the complexity, post-translational modifications, etc. And each one of them contribute to the diversity of the phenotypes. And again, like post-translational, as I said, post-translational modification, the structure and function of the phenotype, structure and function of the protein depends upon the post-translational modifications. see the diversity of modifications that a nascent polypeptide has to undergo before it takes up a role in determining the phenotype. Post-translational modifications of proteins refers to chemical changes, right? And these chemical changes are very vital to determine the functional status of the protein. Of course, that is the basis for a particular phenotype. And now, like transcriptomics, now we have the proteomics, you know these things. Already for last 15, almost 15 to 20 years, this subject has grown, this field has grown to account for all the type of proteins that are present in the body, right from birth to death. That means you could see the analysis of the total proteins, characterization, etc., etc. And like any other, already a human proteome project is in place, and all these things have been done by different pharmaceutical companies because they could correlate with a particular disease. And once they correlate with a particular disease, it is a drug mafia. So as I said, the depth and the breadth of the proteins vary from one individual to the other, from one tissue to the other, and plenty of information is available. You could see that, for instance, eukaryotes are 15,000, bacteria to 3,300, viruses are 42, human proteome is 92. Like that, people are trying to understand the mechanism of splicing and also the diversity of proteins and how these proteins change the phenotypes. And now, an important step in the phenotype, realizing a phenotype, is metabolome and the metabolomics. Biochemical composition of the cell is the metabolome. And now metabolomics like genomics, proteomics, etc. Metabolomics is the measurement of all the metabolites in different tissues now. Concentrations of metabolites at a given point of time. And again, people have profiled the metabolic profiles of different tissues, different organs during development and differentiation, during normal phenotype and a disease phenotype. So a metabolic fingerprint could give you a hint, a forecast what type of normal development it is going on, if not what type of disease that is likely to happen. So like DNA fingerprinting, today you have metabolic fingerprint, and that is related to a particular phenotype or a disease. Metabolic fingerprint is nothing but concentrations of metabolites at different points of time in different spaces of the body. As I said, 50 different human disease phenotypes can be directly related to the fingerprinting. So it's highly individualized medicine, personalized medicine today. By looking at the metabolites or metabolic profile, people could correlate. Already the data is available for 50 different human diseases now. That means if disease phenotype could be taken directly from, if you have the technology, by analyzing the metabolic fingerprinting. Again, see, already the companies have invested human metabolome database because the rewards are very high once you have such database, and based on that, you can develop many other preventive or the drugs. Of course, like this, I can go on and say how the phenotype is going to be determined by and looking at the entire system instead of looking at one gene, one protein, or one cell. Holistically, one has to look at, and that is taken care of by the entire system. Always the temptation is white eye gene is present, therefore it gets white eye. That was how we started. But today it is not as simple as that. Even though white eye gene is there, to get a white eye, a whole lot of things have to happen during development. I will show you now. Let's click. And all these omics now, from genomics, epigenomics, proteomics, transcriptomics, metabolomics, etc. How do all these fit together to give you a phenotype? That is the challenge. And there is a new template, and that template is called Babel Library. is a masterpiece of the famous, it is based on a concept of the library. So all these databases, all these omic databases are available at one particular place, right? And it is based on the concept of library. Bibliomix is an integrative web-based platform for functional analysis of transcriptomics, proteomics, genomics, etc. So you might have, today robots can develop all this data, but somebody has to sit and develop softwares to have relationship to get a meaningful phenotype. So Bibliomix, already 7th edition has come now. As and when more and more information is coming, 7th edition is available, where all this data is available in one place, and people are getting busy in analyzing it to get more and more information about the diseases. So this is the flow. You could see genome, the epigenome, transcriptome, proteome, metabolome, and the phenotype. At every stage, this is crucial. The environment where we live, how we live, would change all these different things before the final phenotype. So you may have the right genome, but you may not end up with the right phenotype. And this is the genetic network, flow of information. This is the phenotype, the layers of information. So this is the genome. And by the time a phenotype is going to be realized, you could see all types of intermediary things have to happen. It could be transcriptomics, proteomics, interactomics, epigenomics, metabolomics, glycomics, and all these things have to happen to get a phenotype. Earlier, we started, if the gene is present, you can get a phenotype. But the performance of that gene has to go through this circuit, and then only you can do this. So the genotype-phenotype relationship is not as simple as the equation I wrote in the beginning, and today, and this network is becoming more and more complicated now. So this is from an ophthalmology journal, but this particular phenotype, and I've modified it in such a way that this flow of information have to happen to get a particular phenotype. So the disease can be cropped up either this stage or this stage or this stage. So thereby what is happening is highly focused drug development is possible because you can detect the defects in the phenotype at any one of these stages. It need not be here, it need not be here. All these stages have to go as expected, then only a normal phenotype can come into picture. So the genotype-phenotype relationship is extremely complex today, and this is like a circuit. This is how the things are. Already we are in the synthetic biology 21, and it is moving forward to give us new insights. See, this is the realization. After doing all these things by enabling comprehensive assessment of genetic mechanisms leading to different phenotypes for the same mutation in two different genetic backgrounds, he says that it is the genotype-phenotype is extremely a complex phenomena. It is not as simple as we thought so far. And like that, from 2010 onwards, I can give a series of references to see, and now people are realizing that genotype-phenotype is not as simple as we started with Caenorhabditis elegans or Arabidopsis or Drosophila, particularly in human genetics, because all us people are wanted to establish the relationship between genotype and the phenotype, or the genes and the diseases. Things are moving on very fast to realize how complex this relationship is. Because when we teach, we'll simply say genotype and phenotype, but underlying mechanisms are extremely complex. With the development of human genetics and computational biology, as well as bioinformatics technology, during the past 2 decades, people know more and more about the genotype, transcriptomes, and phenotypes, as well as the nexus. That means to become a holistic biologist, one need to know many things before we decide whether it's a normal growth, normal development, or an abnormal one. Last, I will take on disease now. Genetic interactions identify combinations of genes and variants on a phenotype. See, in the net genetic circuit I showed you, genetic interactions are very crucial in different combinations, to decide the phenotype. Of course, now they have done extensive work in the budding yeast. I will not go into the detail. Budding yeast to show by changing the genetic network, by genetic background, you can get a different phenotype. New insights into the genotype to phenotype relationships have been worked out in the budding yeast by changing the network. So particularly these things are extremely useful when we discuss epigenetic basis of diseases now. So in budding yeast, Saccharomyces, it has given us a lot of clues today to understand how genetic network is crucial to guide a normal development and also to commit some errors to lead to a abnormal development. Again, this is another syndrome. Even in this syndrome, all these symptoms are there, and the expression of this Engelmann syndrome, this is a complex disease, and this complex disease, all these symptoms are there, like severe developmental delay, movement or balance disorder, ataxia, behavioral uniqueness. All these symptoms are there. It is a syndrome, as I said, speech impairment. And if you look at the genetic basis for these symptoms, very recently they have published this this particular age syndrome could be due to a deletion in this region or uniparental disomy, like that. The phenotype is highly complex and the genotype is much more complex. So I started the equation with genotype to phenotype. It is not as simple. The more and more you study, particularly in complex eukaryotic systems, you get varieties of information, and one has to be extremely careful in establishing the relationship or the correlation, because as the last example I have taken, the syndrome, the efficacy, the manifestations of the symptoms vary in different individuals depending upon the type of damage. So finally, today, what all we studied now is basically algorithms and mathematics, chemistry, biology, physics, ethics, informatics, and engineering. Biotechnology, biogenetic engineering, etc. Not exactly electronics, mechanical, or computer engineering. All these things are involved in biology today. So for all teachers like us, one has to be very clear about the foundations of all these things. Then only holistic biology can be taught and new paradigms could be recognized. The message is, I started with a simple equation, but today, because all these omics, etc., biology, as you have seen, is entering a new era that is data-driven. So all of us have to equip ourselves and have to equip our next generation of students to realize biology is not as simple biology as we taught, as we learned, but it is on a different footing. But basics have to be taught, and basics form the foundation for higher learning. And I hope universities take care of these things. Then only new things can come out of the university campuses. So I don't know how much time I have taken. Okay, 1.5 hours. Thank you very much. Sorry, Bharati. Thank you very much. Sorry, I took a little bit more time.
Very good. Thank you very much, sir. Very long lecture, but it is useful for the students, and we have a paper of molecular genetics by putting all these contents in the syllabus. Whatever is left in the syllabus, I am telling these people to incorporate whatever you are explained so far. Everything is not there. So some aspects are not covered in the syllabus, and in future course of time we are trying to incorporate all these the last slides what you are explained.
Niranjan, my point is not to include everything, but we must teach foundations as clearly, concepts as effectively as possible.
Yes. Once these all things come, then concept will come.